Good morning, everyone, and welcome to the third in our series of Symposia in collaboration between the National Biofilm Innovation Centre in the UK and the India Biofilms Society. We've got a very packed agenda today with 8 speakers, each giving us a short presentation about their area of research and where there may be opportunities for collaboration. And we'll be alternating between myself, Will Green, also from the National Biofilms Innovation Centre, and either Snehal Kidam or Karishma from the India Biofilms Society to introduce the speakers. We would ask that you put questions into the Q&A box you should see on your screen, rather than in the chat. And would you please put the name of the presenter you were asking as well. You can ask questions of any of the presenters at any point during the morning or the afternoon in the case of India. So without further ado, I'm going to hand over to and introduce Maria Salter, Dr. Maria Salter from the University of Portsmouth, who's going to talk to us this morning about marine biofilms on man-made surfaces. Maria, over to you.
Hello, good morning, and thank you, NBIC, for inviting me to give a talk on this, to presenting these lovely people, and happy Saturday as well. I'm saying this as I'm preparing to share my screen. If you give me a moment. All right. Okay. Okay, so good morning again, or good afternoon. Today I will be talking about marine biofilms and my lab's interest on their attachment on man-made surfaces in the marine environment. So marine biofilms, all of you, I'm aware that you will know what biofilms are, but I'm going to slightly introduce marine biofilms because they're slightly different. So first of all, they are very, very complex. And this is the process that takes place in the marine environment when you have an initial attachment of bacteria within seconds of exposure. And then you later on, you will have the incrustation within the EPS and the biofilm formation and recruitment of other microorganisms like diatoms and other protists, and following up with a culmination of other larger macroorganisms like microalgae larvae and so on. This process may take from days to months, depending on the environment you are, depending on the season you are. So it's a very, very dynamic and very, very complex system. And that differs sometimes from other less environmental biofilms. So biofilms, just to highlight, they actually always precede microorganisms. And let me actually change my pointer so it's a little bit more obvious what I'm saying. So they precede microorganisms, and actually there is a lot of evidence saying that they attract or they have an interaction with microorganisms. So a very interesting point to look at them in any case. And they use chemical cues sometimes to attract them, and also they act as a food source for larger for larvae and so on. So why is this a problem in the man-made surfaces in the environment? I'm just going to give you some examples. If they attach on ship hulls, the biofilms themselves can cause penalties in fuel consumption of up to 18%, even if they are 100 micrometers thick. So this is a huge amount for just a tiny layer of biofilm, and up to 40%. And I think there's a larger number there associated with that, with microorganisms. So over the last few years, it was calculated that anti-fouling technologies, which these are technologies deter microorganisms mainly, they provide the shipping industry with 60 billion of savings per year, and they help reduce 384 million of tons of carbon dioxide per year. So extremely important. So I have these two images. On the left, you see a biofouled hull with microorganisms, and you can see if it's a tanker or a large ship that has a very specific timeline, the issues that will cause in fuel consumption and emissions. And on the right here, you can see the biofilmed hull that is on a technology that is namely called far release coatings. They are really good at stopping microorganisms, but the biofilms are mainly and always quite there. So the ships has to be removed and then be cleaned. So huge issues. The International Maritime Organization, they came up with some new kind of strategies, and they want to reduce CO2 emission by 40% from the 2008 levels by 2030, and by 50% in 2050. So there is some urgency there, trying to understand and stop these processes. Other issues, here is on the left, this is a submarine, obviously not very, is suffering from lots of growth. This is in aquaculture, so you can see the nets that they are fully covered, so there is no exchange of oxygen, or not so much, and then whatever they are supposed to protect is not actually happening. You can see some pipes here underwater that they can actually impact the integrity of the material. Some turbines here that they are in the environment, wave turbines, I believe, and these actually, as you can see, they're heavily fouled, and sensors that they are exposed in the marine environment to measure the health of the oceans are also being impacted, and therefore, the measurements are not always, they have to be taken into consideration. So a lot of issues with all these industries that they are impacted. And also, don't forget microbially induced corrosion that is largely impacting platforms such as in the oil industry. Let's talk about also toxicity that comes out from current anti-fouling technologies and past anti-fouling technologies, specifically like TBT, that used to be known to be the worst chemical ever introduced in the environment, and it was banned completely in 2008. And then you have the translocation of alien species from port to port within the shipping industry. So these are all issues that they have to be combated. So this is what my lab is doing. So we are focusing on biofilms, environmental biofilms, with focus in the marine environment. We're looking at preventing methods. We're looking at understanding attachment, and also we're looking at the environmental aspect. So how are biofilms going to be impacted by climate change, and what is happening with biofilms that attach on plastics and microplastics? And I'll give slightly few examples of each of these areas. So initially we start looking at prevention, looking at natural products that they could help us understand stop biofilm formation. And we looked at several extracts, crude extracts, and found that, and these were tested against marine biofilm forming model species. And we found some really good candidates like jaguarone, which is the black walnut. And when we incorporated this compound into a surface, a model surface, it indeed, as you can see here on the left, this is where it has the compound, and on the right, this is the control, where you can see a full grown biofilm within, I think it was 24 hours. So these were some good candidates. However, with natural products and products in general, it's very difficult to put them into play, because you have the legislation, very strict legislation. So currently we're looking at biomimetics and bio-inspired approaches. So mimicking the surfaces of other marine organisms that look, and they are very successful. So please do email me if you have any questions about this. Follow on, the other thing we want to see is what happens at the interface, what happens at the very start of attachment. So in this case, because the marine environment and many other environments, not only the marine, are very dynamic and there is flow. And often in tests, this is kind of put in aside, it's not really looked at. So here, I took the same natural products and I put them on a microfluidic device. This is nothing, and this is a channel that has the natural product, and this is a channel that doesn't have the natural product. So I'm going to put, hopefully this will work. and I will play this video. And you can see the same thing. This was a 14-hour experiment, and you can see that in solution, bacteria start to attach, but not much of an attachment takes place except on the walls. So we can see that the cells are elongated, therefore trying to replicate but cannot, and there is a systematic impact on this. And then you can see on the bottom that a full-blown biofilm is grown when compared to here. So very interesting to look at as a model. This is from a commercial product that I bought, that we looked at the moment, a microfluidic device. But later on, I designed my own microfluidic system that you could actually incorporate surfaces in, because this one you could only look things in solution and a very low shear stress. So we designed a new microfluidic device that could take into account the flow conditions that take place in the environment, very high shear stresses. And these were all computationally modeled and understood what is the environment within the microfluidic device. And this is some images like this, as you can see, from confocal laser scanning microscopy. And if we understood when we did the 3D construction of this, of the biofilms, the typical pores that you know that exist, they did exist. And these are the low shear stress. There is no compound here, just bacteria growing at different shear stresses. This is low stress and this is the highest. And you can see that the high shear stresses bacteria do not, this is actually a 4-hour experiment. Only small colonies are being produced there and at and quite very compact. While here on the low shear stresses, a very different behavior. So this should be taken into account when we're talking about experimentation. And how things and the flow actually does help and does have an impact of how the compounds we're testing are distributed within the biofilm. So the other thing that we are doing is actually, okay, you have a technology that you think it works in the environment, in the lab, but what happens in the environment? In most cases, things don't work in the environment. So in this instance I'm showing here, I have a little rosette where I incorporate different surfaces. I'm going to put my pointer back on, different surfaces, and these usually you can coat with whatever you want. And in this case, I have some far release coatings model, and they are commercially available, and I expose them in the environment. I have exposed them in Southampton, actually. I have exposed them in Portsmouth, in Italy, and so on. And this is another wider scale experiment, because here we just wanted to extract DNA and do microscopy, but here we needed RNA on the right, so we needed a larger surface area. So in this case, we expose our material and we do further omic approaches and to understand both the diversity, but also the functionality of these microorganisms, of biofilms, early stage biofilms, I have to say. Sorry, I'm moving a little bit fast because I have to cover too much. Another project that I'm currently looking at is how will biofilms respond to different CO2 conditions? And this is a large scale experiment that we designed a few years ago. Actually, it was it's led by the BioICID project, and is by Guillaumard and Kiel. And in this case, we introduced different just inert surfaces, glass, into large scale microcosms, and you can see here how big they are, and these were exposed to different CO2 conditions from the surrounding water that was filled with upwelling nutrients. And this is a study that I'm actually in the process of publishing at the moment, with very, very interesting results, and the first of its kind. And finally, I'm going to finish with the current project, where there is a collaboration with actually the Natural Environmental Research Council and the National Research Foundation from Singapore came together to look at the issues with microplastics in Southeast Asia. So there was a 6 million investment where 4 projects, including ours, was included. And we, in this process, actually just kicked off a couple of weeks ago. We're looking at microbial transformation of plastics in this area. And the questions that we're going to be answering is investigating the modification of the surfaces in those environments. Research the hazards and risks and the impacts of the plastics in the environment, and intervention and mitigation of these plastics, because we are collaborating with Center of Enzyme Innovation, that they have, I don't know, it was quite a lot in the news the last couple of years. Our colleagues have modified an enzyme that is eating plastic, and this is going to be part of this project. And these are our collaborations. So
Maria, your time's running up, I'm afraid.
Yes, this is the last slide.
Okay, great.
So this is Vietnam. This is some of our collaborators: Vietnam, Singapore, Indonesia, and Philippines, the key collaborators in this. And of course, the whole consortium that is now put together, all four projects it's extremely interesting to collaborate. And this is my last slide. So I hope I didn't run too much. And thank you again, NBIC. And this is some of my students. I didn't have time to find more of pictures of my students and my lab. So thank you again for hosting. And please, please feel free to ask any questions. Thank you again, Mark.
Thank you, Maria. Thank you for a wonderful, sweeping overview and some detail on that important field of marine biofouling with some phenomenal statistics. So there was a question for you in the chat, which maybe you could address directly with the questioner. So without further ado, I'm going to hand over to Snehal to introduce the next speaker.
All right, sure. Go ahead, Snehal. You can introduce the next one. Sure.
Yes. Okay. Thank you. And our next speaker is going to be Dr. Dikshit. He's going to talk to us about his group's works on biofilms as well as antimicrobial resistance in enteric pathogens. So I'm just going to hand it over to you, Dr. Dikshit.
Thank you, Dr. Sneha. Okay. Good morning and good afternoon, one and all. Today I'm going to talk about some of our work, what we are doing on biofilms as well as antimicrobial resistance, especially in enteric pathogens. When it comes to enteric pathogens, first comes to our mind is mainly Salmonella and E. coli. Basically, these two are the organisms which we are working with. We have studied certain aspects of Salmonella which we have isolated from environments such as seafood or marine environments. So I'm going to talk about more on antimicrobial resistance as well as biofilm work which we have done using these multi-drug resistant strains. So I will not explain all these things because it is well known that Salmonella is an intracellular pathogen capable of infecting microphore cells in the enterocytes using their pathogenicity. And similarly, you have different pathotypes in E. coli with different virulence capacity. Some of them are capable of forming thick biofilm in the enterocytes. Now, all these pathogens are also capable of showing drug resistance with different mechanisms. For example, some might have drug inactivation by producing penicillinase enzyme. Some shows decreased permeability, or change in the receptor might not allow the entry of antimicrobial agents into the bacterial cell. Some will have active efflux pumps, which mainly facilitate the efflux of antimicrobial agents. Some will use alternative pathways. So with this background, we have identified some of the multi-drug resistant strains carrying integrons. These isolates were mainly obtained from seafood, especially Salmonella veltivirden and Salmonella Newport. We identified integrons, which are mobile genetic elements mainly present in plasmids carrying co-triamoxazole resistance with certain ORF gene. We have also identified certain strains carrying silent antimicrobial resistance. That means they carried antimicrobial resistance genes without showing phenotypic resistance. And later we found that these genes were not complete. Promoter regions were absent. That was the reason why they were showing why they were not showing any phenotypic resistance. This was published later. Then we could also sequence the whole genome of one of the MDR strain, that is Salmonella veltivirden. These are all non-typhoidal Salmonella serovirus which we are talking about. And we found that there were 109 genes responsible for resistance or toxic compounds, or several multi-drug resistant reflex pumps have also been identified. Further, when we performed a pulse field gel electrophoresis, we could separate these two strains which are MDR, like Newport as well as veltivirden, when compared to other strains which were grouped in different clusters, whereas these two MDR strains were we could not group in any of the clusters. They stand out separately. We have also developed some of the assays to detect drug-resistant genes or virulence genes for simultaneously detecting these genes in a pathogen like Salmonella, which is also a foodborne pathogen. And we found that this assay, multiplex PCR assay, could detect Salmonella from food sample. The concentration was around 1 nanogram per ml of original dilution. This was also later published. Further, we identified certain mechanisms which are responsible for fluoroquinolone resistance in clinical as well as environmental E. coli. Now, fluoroquinolone resistance can be due to many mechanisms, maybe point mutations in the QRDR regions, that is quinolone resistant determining regions, may be responsible for its resistance behavior, or they might have plasmid mediated quinolone resistant genes, or they might show overexpression of efflux pump. But among all these, mutations or point mutations in the QRDR regions are important for showing quinolone resistance in these pathogens. So identifying these point mutations is very important. We have different techniques to identify these mutations, especially RFLP technique as well as sequencing. But RFLP can be done only if there is a restriction site in these regions. Whereas sequencing cannot be performed unless it is very important. Since there are multiple mutations in these regions, for every gene, if you perform a sequencing, it is a costly effort. That's why we have developed a assay called mismatch amplification mutation assay, which is similar to allele specific PCR, where we design a reverse primer with a single mismatch at the 3' end. So if there is a mismatch in the original gene, this particular primer cannot extend. There will not be any amplification. So in a resistant strain, we will not get a band for the reverse primer, whereas sensitive strain will show up 2 bands when we perform a polymerase chain reaction. So this was developed, and it is important when we have large number of samples and when we want to know status of QRDR mutations in these isolates. So all these isolates were also subjected to biofilm formation, especially in vitro gut condition. Majority of these isolates were known to form biofilms, especially MDR strains. As we all know, biofilms, again, a previous speaker has explained how it forms. But these biofilms are also an important mechanism for microorganisms like enteric pathogens, such as Salmonella and E. coli, to withstand effect of antibiotics. So antibiotics cannot percolate these biofilm layer when there is extra polymeric substances present in these layers. They will prevent the entry of these antibiotics. So a sensitive strain can also become resistant when there is a biofilm. So this is what we have studied in our isolates. So we have conducted the antimicrobial susceptibility assay for 59 non-typhoidal Salmonella serovirus. And we found that majority of the serovirus were resistant to either ampicillin, nalidixic acid, tetracycline, or chloramphenicol. Now, all these isolates were also subjected to biofilm production using a normal microtiter plate method, which is a crystal violet based assay. And these isolates were also subjected to bile, that is 3% bile. Bile is one of the infection related gut condition among many gut conditions which can influence biofilm production as well as virulence of any enteric pathogen. That's why that was the reason to use bile as an important stimuli here. And we found that majority of the isolates which didn't show strong biofilm formation in control condition did show biofilm when we exposed them to bile. So that was observed here in the case of Salmonella, especially even 42% of Salmonella strains which did not form biofilm in control condition reduced it to 14% in the presence of bile. So you can see here in this table, some of the isolates which were non-biofilm former become strong biofilm former in the presence of bile. So there is a triggering factor here which triggers the production of biofilm in these isolates. This was further confirmed by chalcophore staining as well as Congo-Reid method. We also saw a difference in growth rate among these isolates when they subjected to bile. Bile was able to reduce the growth pattern of these isolates or growth of these isolates, but they allowed them to form a strong biofilm. Further, we studied the genes responsible for biofilm production, their expression, of course, by real-time PCR, and we found that genes like GCPA, which is responsible for biofilm production, was expressed more when compared to other genes. Similarly, a negative regulator, CSRA, which was showing lowered expression, it is mainly responsible for motility and female production in the case of motile organisms like Salmonella. During biofilm formation, what happens? Most of the microorganisms are in sessile form, so they need not have motile nature or female expression. That's why this particular lowered expression was observed in this case. However, other genes like genes related to quorum sensing and genes related to stress response did not show much variation, or they showed variation among different strains with different classification. So this was observed in Salmonella. Similarly, we have also subjected all our multi-drug resistant clinical E. coli strains as well as environmental E. coli strains to different gut conditions. Not only bile, we subjected them to NaCl, low iron, high iron, pH, or temperature. We found that majority of the isolates became strong biofilm former when we subjected to these gut-related conditions. For example, bile, you can see the increase in biofilm formers from 8 to 24. Similarly, in the case of pH, when there is a variation in the pH, they show strong biofilm formation from 8 to 23. Similarly, in the environmental isolates, although there was not much variation, some of the strains did show strong biofilm formation in the presence of bile, whereas other gut conditions did not have much effect on biofilm formation in these isolates. So finally, I could conclude my talk by saying that irrespective of their resistant pattern, the bacteria can form biofilm based on their environmental cues to which it is expressed to. And certain host immune factors, like innate factors, these bile or variation in the gut pH, can suppress the growth, but then yet they bring out a persistent strain, which can withstand harsh environmental condition. Similarly, close proximity of these isolates within the gut in the form of biofilm matrix can lead to transfer of antimicrobial resistant genes as well as virulence trait if they are present in movable elements like plasmids. And especially bile can act as a signal for Salmonella to turn on genes involved in biofilm production and to survive in such an harsh environment. So our future work will be mainly to identify mechanism that triggers the biofilm production under these conditions. So this was later published, and these are the people whom I can acknowledge. Thank you. Sorry for going very fast because there were many slides.
It's all right. Thank you so much, Dr. Dixit. I think we can if there are questions, you can find them in the Q&A function or in the chat box. And you can, I think, just type your answers in and we will move on to our next speaker.
Thank you.
Thanks very much. So I'd like to take the opportunity to introduce Chenny Chang of Newcastle University to give a talk on understanding and harnessing bacterial population behaviours. Thanks very much.
Right, let me share my screen. Hi everyone, my name is Chenny Chang. I'm original from Taiwan and now I'm the lecturer in the School of Data Science in Newcastle University. And many thanks for the opportunity for sharing my research interest. And I have a very broad research interest. I did my PhD in Nottingham with Paul and Miguel, and I also done some very unique predatory bacteria. These are small bacteria eat larger bacteria. And also I collaborate with my Malaysian collaborator to study the environmental bacteria and also work with a clinician to study the epidemiology of the group B Streptococcus. And today's talk, I mainly focus on the cell communication and of course the anti-biofilm materials discovery. So I'm sure you all know bacteria can talk and listen to each other, and this phenomenon we call quantum sensing through the small signal molecule. And there's a three component important for quantum sensing. This is including the quantum sensing signal, including the synthesis, including the responding protein. And when the bacteria sense the signal, they will change their phenotype accordingly. So this phenotype including several different variants, secondaries, metabolism, of course, motility, biofilm, and etc. etc. And the idea is if we can reduce the quantum sensing signal, or we can interrupt the signal synthesis or responding protein, we can change the phenotype. So no more variance factor production or no more biofilm. And this approach we call it quantum quenching. And for this area of the research, I aim to find the new compound which can interference their cell communication. So I present here the two study. The first study is using the Pseudomonas aeruginosa. This is opportunistic pathogen, and they have a very complex quantum sensing network. And after screen 127 nature product, we find there's a interesting one which can inhibit the signal synthesis. And from that time, and I think we are the few lab to identify this small molecule inhibit the signal synthesis. And of course, it's not only interrupt the quantum sensing activity, it also inhibit some Pseudomonas variants production. And later, we try to understand how this small molecule interact with this protein. And the second research is using the Chromobacter violacum. And if you are in the quantum sensing field, you may know the Chromobacter is a very famous and popular quantum sensing biosensor because they produce this purple pigment, and this purple pigment is controlled by the very simple and typical quantum sensing network. And in this area, we didn't look at the nature product. Instead, we're looking for the artificial polymers, and these polymers are very highly branched, and you can functionize, you can add in any chemical function group on it. And then we screen quite a lot different high branch polymer, and we identify one is very interesting, inhibit the quantum sensing activity or quantum sensing controlled violacum pigment production. And later we are trying to understand how this highly branched polymer can interference the quantum sensing. And the second area or second research one try to talk is about biofilm. And I always think biofilm is the city of microorganisms. And we all know biofilm is almost everywhere, from the environment to the industrial pipe. And in a clinical setting, biofilm is a big trouble because biofilm can block the catheter, or they can form the biofilm on biological surface like our teeth. And they are not just 2D flats, they are actually the 3D, just like our city. There's a 3D structure over there. And we all know this is the bacterial biofilm typical model. Bacteria or planktonic bacteria swimming around, floating around, attached to a surface, and then they grow, they aggregate, and forming this microcolony, and eventually form this mature biofilm. And the idea is, if we can have anti-biofilm or anti-attachment material to stop planktonic bacteria attached to it, so there's no more microcolony formation or biofilm formation. So to address this approach, we develop a polymer microarray technology. And basically, we have a lot different monomer, and by mixing this monomer in different composition or different level or ratio, we can create thousands and thousands different polymers. However, to screen those polymers is almost impossible because the amount is too much. So we create this microarray technology. And from here, you can see each tiny spot is each different polymer. So they have a different surface. So we can challenge this surface using 3 fluorescent labeled bacteria, and we can screen this polymer in one go very quickly. So the screening goes through several rounds, and we eventually identify few of them have anti-attachment and anti-biofilm properties. And then this property not only in this tiny small spot, it can also scale up and the coating on the catheter. And then we even put the coating the catheter to the mice to do the animal model. And you show after 4 days, the mice with the coated catheter can reduce the bacteria bacterial infection and also reduce the bacterial colonization. And this is a very interesting research. And of course, we not stop here. We try to understand how this polymer can do this thing, can anti-biofilm or bacterial attachment. So we've done quite a lot surface chemistry analysis, including the water content angle, roughness, and the element on the surface. And of course, we need to know how the bacteria responds to this. Is that the polymer kill the bacteria? And we try to do this growth curve to see if the bacteria growth inhibit in presence of this polymer, or what's happened, the surface can interference the life and death of bacteria. And in the future, of course, this is a very interesting. We try to understand how this bacteria responds to so many different polymers, and we want to identify what is the key sensing mechanism for this bacteria to recognize so many different polymers. So today I share 2 areas of my research. One is to looking for the new compound interference, the bacteria cell communication. And we hope to develop some virulence regulation or antibacterial infections approach against bacteria under this antimicrobial resistance crisis. And the second area, we try to understand how bacteria responds to different surfaces and understand their controlling mechanism, especially in the initial state. And a few years ago, I've been to India, IIT Kharagpur for a conference, and I really love it. And I would like to invite everybody come to beautiful Newcastle and visit our beautiful city campus, and of course, come to the dental school, and we can share the idea and work together in the future. Thank you very much.
Thank you very much, Jenny, for that very interesting talk. I'd like to hand over to the Indian team to introduce the next speaker. Thanks very much.
Thank you, Will. I guess I'm audible. Yep, and the repair activity in my house is not audible. That's also a good sign. All right, so our next speaker is Dr. Karthik Balasubramanian, a friend and colleague in the biofilm world in India. And I can take a little bit of credit by saying that we live in the same city in Western India, in Pune. All right, so he's from Agarkar Research Institute. Over to you, Karthik. Welcome.
Thanks, Karishma, and thanks for the biofilm community, both in India and UK, for inviting for this talk. Okay. So I will just, my talk is something related to the first talk we had on the marine biofilm, but I work mainly on freshwater. So my talk is on how to use the riverine environmental biofilm to assess the river health in biodiversity hotspots. So I will start with defining these three key words which is used in my presentation. First is the river biofilm. I mainly concentrate on epilithic biofilm on the stone from the river, and my focus organism in the entire biofilm is diatoms. I especially focus on diatoms, and the aim of the work is to understand environmental monitoring. It can be the current environment and it can be the past environment. So that's what we will be going to talk today. And in terms of biodiversity hotspot, globally there are 32 biodiversity rich zones across the globe. The Conservation International have defined these areas, which is shown in the left top map. So if you see, my study area is in the Western Ghats, which is along the west coast of India, which starts from the just above Mumbai and it goes till the tip of the peninsula. So these are my our sampling sites. We focus mainly on Western Ghats. So why Western Ghats? Again, it's one of the highly biodiversity rich place on Earth with literally new species coming every other day, not from microorganisms, from big trees to plants to monkeys to birds, and for example, like in frogs, almost we have new species every fortnight, even at the level of new family also comes from Western Ghats. So that's the reason why we study Western Ghats. And then it's also water tank for almost more than few million people. So it's highly threatened ecosystem. So that's why we study this. So when we take a cross section of Western Ghats, it's a just an art we did. So it's close to the coast, the Arabian Sea. So the one side is with a lot of rainfall, the other side is with less rainfall and a lot of disturbance. So this is the cross section of the Western Ghats mountain system. So we in Pune, it's on the eastern side, and some of my colleagues here from Mangalore are from the western side of the Western Ghats, so with heavy rainfall. So my study organisms are diatoms. They are microscopic algae and photosynthetic, made up of silica cell wall and very species rich. The known species are more than close to 50,000, and the number of predicted species in diatoms are close to 2 million species are yet to be described. And the beauty of diatoms are they are environmental specific. They can indicate change in pH, conductivity. Almost every physical and chemical parameter in the river can be determined by diatom. On top of it, they also contribute almost 25% of global oxygen. So, we collect diatoms from biofilms from aquatic and semi-aquatic, even in terrestrial habitats, and including extreme environment also. Here, we mainly concentrate on biofilms from rivers, and then moss patches, and then ephemeral pools on the mountain tops, frozen lakes in the Himalayas, and then acidic swamps on the Western Ghats, and then the wet walls along the mountain passes. So these are our main study area. For this study, which I am focusing here, we mainly focus on the biofilm growing on the riverine sites, mainly stones and plants and sediments. So what we do is we collect the biofilm, and then we are interested only in the diatom. So we oxidize everything else in the sample. So we burn we boil the content with the samples with nitric acid. So we get just a pure silica, just the items for identification. Along with that collecting, the item collection, we also take water samples to measure the water chemistry parameters like pH, conductivity, and nutrient, ion, and cations. So from each sample, we count 600 individual diatom. We make a permanent slide from each sample, and from each sample, we count 600 individual diatom, and then we calculate the relative abundance data from for each sample. And when we are doing this, globally, diatoms is one of the least studied group of organisms. So almost 30 to 40% what we see are new. So we end up describing so many new species and even many new genera also we describe. Most of our expert goes into identifying the organism and describing the organism. And then in this work, we focus only on 112 sites. This is the principal component analysis, where this which uses all the water chemistry parameter. Based on the water chemistry parameter, we could categorize the sites on the heavy rainfall western slopes, which is highlighted here on the green square, and the sites on heavily disturbed less rainfall zones on the eastern slopes of the Western Ghats, which is indicated by the red color rounds. So these are the two major gradients which determine the nature of the rivers in the Western Ghats. And in terms of diatoms, what we found is like in 112 sites, we counted 67,000 individuals, and we classified the diatoms based on the pollution gradient and the altitudinal gradient. These are the two parameters which is determining in this canonical correspondence analysis. So based on this, we classified the diatoms into three groups, whether they occur in oligotrophic, low nutrients, and then mesotrophic, moderate nutrient, and then eutrophic. So this classification, we use it to identify the status of rivers and streams of the Western Ghats. And the same analogy, we also use it to the sites to understand the past environmental condition. For example, this study site, which is like hardly 100 kilometers from Pune, it's a place called Kars, which is known as the Valley of Flowers of Maharashtra. This is the particular state here. So where we took a core of 2-meter core, and we studied the diatom deposited in the sediments. So based on whatever the environmental preference we arrived at based on the modern samples, we use it in this, and then we conclude like, for example, if you just pay attention to this green and red, the concentration of these two species, this green species is here since last 8,000 years. We did a carbon dating, so we know the exact age of the sediments. So this particular species is living here since 8,000 years until very recently, when humans started using this lake. So this pollution tolerant species started popping up, and then this pollution sensitive species, it got wiped off from this particular place. So from our studies, we came at least like 1,000 species, which is need formal description, and eastern slope and western slopes are the major determining factors for the diatom community on the riverine biofilm. And there is a huge shift in the diatom community in terms of historically in the Western Ghats. And we are also using recently molecular methods to identifying the species. So we culture the species and then extract the DNA, and then based on the RBCL, like we work on the species taxonomy. And in terms of biofilm relevant questions, we also started working on understanding the bacterial community associated with the diatom. So each diatom have its own bacterial community. So we are trying to understand that. And also we are looking at some of the diatoms even now have endosymbionts, like mainly blue-green algae. So we are trying to study those endosymbionts associated with diatoms. And with this, I conclude. Thanks to my funding agency and our lab for doing this work. And thank you so much. I'm ready to take some questions.
Okay, thank you, Karthik. I think you can check the chat window and the Q&A box for some questions when they come in, and I will hand it over now to Mark to introduce the next speaker.
Thank you, Snehal. Thank you, Karthik. That was a fascinating talk and some beautiful images. So it's my pleasure to introduce Dr. Selva Athena Royanan from the University of Edinburgh, which we're confident is the most northerly of our presenters today. And he's going to talk to us about the area of 3D printed bioreactors. So Selva, over to you. Selva, you're on mute.
All right.
Thank you.
Hello, everyone.
I get to say that one time today. Thank you very much.
And hope you're having a productive weekend today. I'm going to talk about my postdoc project that involves 3D printed bioreactors for biotransformation reactions. This project is funded by Industrial Biotechnology Innovation Centre at Scotland. It's a collaboration between Recircle Limited, a company that focuses on devulcanizing rubber tires using bacteria, and the fantastic University of Edinburgh. In this presentation, I'll be talking about bacterial biofilm bioreactors for a biodesulfurization reaction. So I'll start with what is known. It's known that immobilization is a very traditional methodology for bioprocesses. And a development of that is the biofilm bioreactors, where stubborn biofilms are formed on plastics or this packing materials, which are either suspended or fixed, and the bioprocess is carried out on them. I'm not going to do much the details of this. What is unknown is if these kind of packed systems could benefit from having a perfectly ordered structure. By the word perfectly ordered, I mean to have a specific geometry that is that's maintained throughout the length of the packing. And this could be achieved by using tri-ply periodic minimal surfaces. The image on the right shows you some examples of the TPMS structures. These structures have complex geometries, and they can be defined by a formula, and they have unit cells which can be multiplied in 3 dimensions, and structures of any desired dimension can be built. The idea popped up based on recent findings that in chromatography, having a perfectly ordered packed bed helps to attain a higher efficiency than a randomly packed immobilized materials. So we wanted to try if this can be applied for a packed bed reactor, which has got the biofilms on it. And we wanted to test it on a biodesulfurization process, which is about the removal of sulfur from fossil fuels. Biodesulfurization was my PhD project. And as I moved to postdoc, we blended that with this perfectly ordered structures. Briefly about biodesulfurization. It's a process to remove sulfur from fuels. Fossil fuels contain organosulfur compounds and inorganic compounds. And when combusted, the sulfur in these compounds are released as sulfoxide emissions, which are harmful to the environment. So it's a legal requirement to reduce the sulfur content. Biodesulfurization uses microbes, especially bacteria, to selectively remove sulfur from these compounds. It's a whole cell biocatalysis process. And in laboratory research, benzothiophene and dibenzothiophene are taken as model organosulfur compounds. And bacteria do this in 4 steps. It's a multi-enzyme process. For example, the dibenzothiophene is sequentially converted into 2-hydroxybiphenyl, which is a phenol. The benzothiophene is also converted into hydrophenoxyethanol, which is another phenol. In laboratories, the presence of phenol could be tested using a Gibbs test. When the Gibbs reagent is added to the culture, then it turns blue, which confirms the presence of phenol, which could have come from the organosulfur compounds that's added to the medium. So in this presentation, blue means biodesulfurization reaction has taken place. The biocatalyst that we use is an actinobacteria, a species under genus Rhodococcus, which are particularly popular for their metabolic versatility thanks to their big genome. They have a mycolic acid-rich cell membrane, which renders them a highly hydrophobic nature, and they're able to thrive in oil-rich environment. And some of these Rhodococcus strains have excellent biodesulfurizing capabilities. When we think about 3D printed bioreactors, there are two ways to approach it. One, you can have the cells and the support materials as a mixture, and this mixture can be extruded into three-dimensional supports like Saha et al. Has done here. Otherwise, these can be made into three-dimensional objects using stereolithography, like Dubin et al. Has done here. This is an emerging technology for bacteria, but it's an established one in the case of animal cells, and that's where we call them 3D bioprinting or bioplotting. It's basically an entrapment kind of immobilization. But we decided to go for another way, to have the support 3D printed first and let the bacteria absorb onto them. And for this purpose, we need to optimize the material's porosity and the biocompatibility. The TPMS structure that we decided was gyroid, because it's been shown that gyroids have relatively higher strength, which is suitable for industrial reaction to withstand the conditions there. And also it facilitates the cell seeding among all the other TPMS structures. Going to this method gives us a lot of flexibility in creating the support material. Thanks to our expertise in chemical engineering, we're able to create polymers with desirable properties by changing the composition. We first tried with butyl methacrylate to get a structure that has got a hydrophobic surface. Remember I said this bacteria allows hydrophobic things. And then we also used Maytec, which is a big chemical here, to create a structure that's got a mild positive charge, because bacteria have a weak negative charge. These are the conditions that facilitate adsorption. When creating our own formulations, there are a lot of conditions that need to be optimized in terms of 3D printer. We use stereolithography and especially digital light projection method. And we had different unit cells for these TPMS structures. We did a 2 millimeter one and a 4 millimeter one, and the exposure time to get them in proper shape has to be optimized. And after all this was done, we finally decided to go for Maytec polymer. So these are the gyroids that we obtained, which have a big positive surface. So the workflow involves culturing of bacteria and 3D printing gyroids. They are put together in the sense we perfuse these cultures through these gyroids to get optimal biofilms formed on the surface. And after that, the biomass and the BDS activity are assessed quantitatively and qualitatively, and the 3D printer reactor is assessed for its efficacy. So this work is a preliminary study that was done over a period of just 3 months. After that, this project was put on hold because of COVID, and I just have resumed it. So it involves packing these gyroids into these syringe columns and recirculating them with culture for facilitating immobilization. And the actual reaction was done at a slower flow rate. What we found was that this material supports biofilm formation. As you can see in this same image, we see nice biofilms of the Rhodococcus bacteria from there. I say it's Rhodococcus because of the morphology, the elongated one, which is specific for Rhodococcus. And down below, you see a gyroid that's not got a biofilm, that's a control. And through our tests, we know that upon third or fourth pass through the columns, biodesulfurization reaction has taken place. The phenols are present in the culture systems, as indicated by the thicker color there. So these are preliminary findings. And the limitation is the capacity of 3D printer at the moment. It can do 7 centimeter long gyroid, but whereas when it's actually scaled up, we need to have longer columns. So at the moment, we are just using columns created by stacking the gyroids on top or having columns in series. And there's optimization need to be done in terms of the retention time. And this is just a platform technology at the moment. This can be further extended to any biotransformation reaction. So I would like to collaborate with anybody who's interested in immobilized whole cell biocatalysts or anyone interested in developing novel support materials, because that's one of the field where a lot of research is also taking place in terms of biofilm bioreactors. Thank you very much.
Thank you very much, Selva, for a really well presented and logical story. And I wish you all the luck with your future work. And we'll try and help you with in NBIC with facilitating contacts both with India and the UK as well. So without further ado, if there are any questions, Selva, they'll be in the Q&A or in the chat. So Snehal, I'm handing over to you next. So thank you very much.
Okay, thank you. So our next speaker is Dr. Srinandan, who's from the Sastra University in Tamil Nadu. And he's going to be talking to us about the work his lab has been doing on biofilms. So I'm just going to hand it over to Dr. Srinandan.
Yeah, thank you. Yeah, so thank you. It's a very nice, exciting moment that second time we are having the NBIC and IBS joint symposium. First one was a success. We can see with Karishma and she got already the collaborative fund. I hope so in future also we'll get this kind of thing. So I'll first introduce you to the place where I am. I'm down south in India, and it's Tanjavur. It's one of the historic place where our university is located. For example, it was ruled by ancient Chola dynasty, which ruled for nearly 1,500 years. And there is a very big, beautiful Brihadishvara temple. I invite all of you, if possible, when you come to India, please have a trip over here to Tanjavur. So there's my lab. We work on, it's called the Biofilm Biology Lab. These are my PhDs and some few UGs and PG students who work in my lab. We work on biofilm biology of mainly the Uropathogenic E. coli and Salmonella. And we also have some translational research going on for the pathogenic biofilm. And we also do some detection with engineers. We do collaborations over here in a university, and we come out with some, attempt to come out with some tools, for example, electronic noses, electronic tongues, or anything like that. And now we have also started to develop some tools so that we can isolate the unculturable bacteria, because a lot of cross-feeding takes place in the environment, and it's very important to isolate as it is and to try to mimic the environment. But nevertheless, in this talk, I'll be talking only about the first one, which we very briefly, and I'll be giving you 3 stories which we have at present, and it's a very briefly I'll be touching upon that. And because biofilm is very well known that they have a spatial structure in them, it's also that there will be adaptive radiation, and interaction among them is very important. So when you put under the game theoretical framework, either there will be a selfishness between them. So the fitness payoff will be more towards one particular species, one particular cell type, and there could be mutual inhibition when both of them fight against themselves, or there could be cooperation. For example, the cooperation which I'll show you, there could be public good cooperation, but we are yet to know about altruism. This kind of fourth, there is a very simplistic version of how the interaction in the specially structured by a film can happen. So first I'll tell you the first story very briefly, that is about the Salmonella biofilm. Yeah, Salmonella biofilm, as we know, it's very important because it gives it favors the long-term survival in the non-host environment for the Salmonella, and it's a contamination risk in food processing environment, and it's also tolerant to many of the antimicrobials. So, very preliminarily, what we found was in the biofilm, in the pellicle, when you take this congruent media, the red colored colonies means that they produce matrix and they are able to form biofilm. White color means they are unable to produce matrix and they don't form biofilms. So, in the subpellicle region, there are more white cells. That means they are unable to form biofilm, and the biofilm cells are there over there in the pellicle, but nevertheless there are cells which are also white over here. So what we did was, we also see this kind of diversity in the population in the clinical isolate. So what we did was, we tried to create a mutant of biofilm formation, and we saw that the when infiltration of the mutant which are non-producers, they are present, when they become more and more, the productivity of the whole biofilm goes. For example, over here in the video you can see, when there is a non-producer population more and more, then it weakens the biofilm. It also weakens the biofilm such that even the antimicrobial things like chlorine tolerance or the ciprofloxacin antibiotic tolerance goes down drastically, very significantly. But nevertheless, what we found was it acts as a public good. The matrix which are produced by the producers can act as public good. Over here, you can see the RFE bacteria and the GFE bacteria. The GFE are the one which produce the matrix, and the RFP which doesn't produce the matrix. So they exploit the particular matrix present in the pellicle, and they also try to collaborate in the biofilm. But nevertheless, what we found was, after a series of image analysis of the confocal microscopy, we found that it is actually the cellulose which is acting as a public good. It is not because the predominant components of the biofilm matrix in Salmonella and E. coli, they are cellulose, colonic acid, and curly. But cellulose is the main thing, because with the mutants, when we saw when the non-producing cell subtypes are there, they keep them away from the matrix. So they are edged out in the periphery. They are kept at bay from the microcolony of the producers. And whereas when the cellulose was taken out, we could see that they try to exploit whatever resources can be produced by the microcolonies of the producing bacteria also. So cellulose can act as a double-edged sword, where it can keep the maybe a possible cheaters at bay, as well as provide them as a public good, because there are also other advantages in the subpopulation variants. Next is, I'll go to the story of the E. coli biofilm. Especially we work with the Europathogenic E. coli, and over here what we have seen is, it's very important because around more than 75% and more than 65% in complicated UTI infections, actually UPEC is very much involved, as well as in the recurrent UTI. And biofilm plays a very important role, as well as in the nosocomial infections. Around mostly predominantly it is the Europathogenic E. coli which will cause the infection. So my student Sandeep, what he had seen was, E. coli also UPEC can produce three different kinds of populations in the single biofilm populations. Then we actually labeled them with antibiotics and we did the co-culture analysis and we checked for fitness and productivity of the biofilm. But over here, what we can found was in the earlier thing about Salmonella story, what we could observe was when the cheaters become more, the cells, then selfishness has been displayed. But whereas over here you can see in some of the submerged biofilm, productivity is enhanced very significantly, very highly significantly. We are working on this to know what is the mechanism and why they are actually the productivity is going up. And there's a second, so over here you can see the kind of mutualism occurring between the cells. And now I'll go to the third one, which we recently published. That is, there is also territoriality in bacteria because it was UPEC and Salmonella. Now I'll tell you the third story where both of these together, when they are there, what will happen? It is like the mutual inhibition. For this also we have done the congruent plate where, for example, there's an illustration by one of my student, Punmani, and she had done a very good illustration of this, how in congruent media, when there is a matrix, both cellulose and curly, it becomes a red cell. But when there is absence of curly, it becomes a pink cell, and absence of cellulose gives a brown cell, colony type, and the cell type, colony type will become white when in case of absence of matrix material. And this is a simple method which we, all the microbiologists, it is well known, and we exploit this simple method to find is there some isolate in the environment, because even now, though E. coli has been the workhorse of microbiology from decades, from centuries, in fact, but we don't know how E. coli can survive in the, live in the environment, in the non-host environment. We know most of the things about the genetics and in the in vivo environment, but we know very less in the environment how it is. So, because we know that predominantly it is the biofilm formation outside, so E. coli can have a dimorphic life, that is in the host as well as in the non-host. In the non-host also we believe that it might be in biofilm. So we tried to isolate cultures which could dissolve the matrix of E. coli so that it could be translational. When we saw that, we got some of the isolates which could in fact dissolve the matrix of the E. coli, and surprisingly the isolates we didn't select for, but we just the emerged cultures which will actually dissolve the matrix. When we saw it was all belonging to one particular family, that is the Enterobacteriaceae, which is in the gut. So this family, when we gave, we saw the different antibiotic susceptibilities for them, as well as labeled for some which is not there, and we performed some competition experiments. Something drastically very interesting we found. That is, we found that there's the planktonic cells, for sorry, I have not labeled over here, there's biofilm, and in the biofilm we can see the productivity goes less, and most of all the isolates, including the UPEC organism, they all want to stay alone, not in co-culture. Okay, so they have a mutual inhibition between them. So this is telling us that it is important that they are fighting for the territory. Okay, who will go and colonize first a particular territory, and secondly, there is also
Dr. Nandan, sorry,
yeah.
Will you be able to just wrap up in a minute,
Yeah,
because
yes,
the time, yeah, thank you.
Yes, yes. Yeah, thank you. So this says that the cells populations are predisposed to form biofilm, and this again we have done the quantitative exclusion was shown by Salmonella, and here I'll wrap up. These are some of my collaborators and mentors, and also the funding agencies which our lab is supported by. And thank you. Namaste.
Thank you so much. You can check the chat box and the Q&A for any questions, and we will move on to the next speaker.
Thank you very much. So I would like to introduce our next speaker, who is Tony Moore from the University of Sussex, to talk about the role and control of the alternative oxidase in Candida biofilm formation. Thanks very much.
Okay, thank you very much for the invite, and I will start my talk. My screen tends to go blank for 5 seconds. Don't worry if you see something blank as we go into it. Essentially, what I'm going to talk to you today about is about an enzyme called the alternative oxidase. Whoops, it is not happening. Yeah. Can you see it?
Happen if you, yep.
Whoops. Seem to have lost it. Ah, right. Okay. I couldn't see it. My screen had gone blank. Okay, so what I'm going to talk to you about today is this enzyme called the alternative oxidase. And specifically, I'm going to talk about its role in the formation of biofilms by Candida. I've worked on this particular enzyme for the last 40 years, so I've know quite a lot about it. And quite recently, what we've just done is to spin out a company called the Alternate Oxidase, Altanox Scientific Limited. So if I go to my next slide. So what is the alternative oxidase? Well, it's a terminal respiratory chain oxidase. Essentially, it's found in mitochondria. It plays a key role in reducing oxygen to water. And when I first started working on it, it was initially just associated with plants. But now it's present, we know, in a majority of fungi, protists, and indeed some human pathogens. Essentially, this protein acts as a stress protein. And in parasites, it scavenges oxygen. Now, we now know that a number of human fungal pathogens also express.
So, really sorry, Tony. I don't think your PowerPoint is coming through properly. If you turn off your share screen and when you come to do it again, select the PowerPoint app rather than the screen. It's just frozen on the first page for us.
Oh, is it? Ah, all right. And I can't. I don't appear to be able to stop it. Okay. Well, my screen has now gone blank.
Yeah, and then when you choose to share your screen again, if you select the actual PowerPoint app rather than just your main desktop screen, that should fix it.
Okay. Right. So I'll try and share the screen again. So what is happening here then?
Until it doesn't work.
Multiple screens.
I'm trying to share it there. Can you see it there?
Yeah, there you go. That's moving through now. Thank you.
Not PowerPoint. It's
not moving as PowerPoint.
Not on slideshow.
If I put it on slideshow, it was working earlier. Is that working now? Is that working?
Yeah, I mean, we see the slides, but it's not in screen show.
Yeah, and they're sort of zoomed in for some reason.
Yeah, exactly.
Tony, I'm going to share it. Yeah.
Okay.
Okay, can you do it then?
No, I'll just say next slide.
Thank you, Matt.
Okay, if you go to the next slide or so. Okay, if you leave it there, and then if you hit go ahead onto the next slide, hopefully there. Okay, so apologies for these technical problems, but hopefully we can continue. I'm talking about this particular enzyme, the alternative oxidase. It's absent from humans, and as it's present in a number of human fungal pathogens that express an AOX, then obviously it is now becoming a potential therapeutic target for the treatment of a number of human diseases. So on the next slide, I wanted to show you the location of this particular protein, and this is really a simplified kind of version of mitochondria, and we all know that mitochondria play a very key role in energy production within the cell. And in this particular one here, what I'm showing is the respiratory chain that's present within all mitochondria, and namely it's comprised of these very large proteins, these multi-enzyme proteins, of which the crystal structure is known. And so I'll place those into a membrane. And if we go on to the next slide, then we'll find out that in fungi, they are associated with the presence of a number of what are known as externally located NADH dehydrogenases, which you can see there. And in the next slide, then the protein I'm talking about is the alternative oxidase. So this is present in the majority of fungi, and indeed in the next slide, if you inhibit respiration in fungi, you tend to overexpress this particular oxidase, and then it allows the fungi to continue to respire in the next slide. And hence it can still oxidize substrates, particularly through NADH or indeed through succinate. And in the next slide, you can see in the next slide that essentially what happens under these conditions, then the energy is released as heat. So if we go into the next slide, please, then we have actually crystallized the structure of this particular protein with my colleagues over in Japan, and we published this a number of years ago. We still remain the only laboratories in the world to have the crystal structure. Now, the interesting aspect about the alternative oxidase is the fact that it's known as a self-folding enzyme, so it attaches itself to the inner mitochondrial membrane. So if you actually express the oxidase in human cells, then you can actually introduce the alternative oxidase. What's of particular importance are these hydrophobic cavities that lead down to where the catalytic site is. Now, if we go on to the next slide, we now know that a number of human pathogens contain the alternative oxidase. As I said, particularly in Candida albicans, and more recently in a new kind of type of Candida, namely Candida auris, in addition to it being present in parasitic infections. And in the next slide, the Candida albicans is an opportunistic pathogenic yeast. You find it in gut flora, etc. And it's a commensal organism, but it can become pathogenic in immunocompromised individuals. And it's the most common fungal species isolated from biofilms, either on implanted medical devices or indeed on human tissue. And it's proving to be a particular problem because now normal treatments with compounds such as fluconazole, you find that now there are a number of resistant strains of Candida. And on the next slide, Candida auris, as I've already indicated, this is becoming an emerging serious health threat. Mortality tends to approach 70% in candidemia patients, and it's almost resistant to all of the azyle and polyene conditions. And in the next slide. And so what we've initially been attempting to try and do is to develop a number of specific inhibitors of this alternative oxidase. we know that both in auris and in albicans, that they tend to rely on the presence of the alternative oxidase. And so our spin-out company, the Altanox Scientific, has been developing now a number of specific inhibitors that can distinguish both between albicans and auris. Now, obviously, what we also need to determine is the extent to which any of the inhibitors affect the main respiratory chain, because obviously via treatment in human cells, we don't want to inhibit our normal cellular mitochondria. And we can find out that a variety of these inhibitors inhibit both the isolated enzyme, and if we go into the next slide, we've also developed techniques to determine Candida biofilm formation, and again treated these. And this is I just want to draw your attention to just two of these. This is the effect of fluconazole on Candida biofilm formation, and this is both on Candida albicans, and indeed we've tried it on auris. And you can see now we're developing a number of these compounds which indeed are proving very specific at inhibiting biofilm formation. And on the next slide, we're also been collaborating with a company called Symcell from Sweden, and Symcell has developed a 96 well machine that essentially can measure total metabolic activity, both as planktonic cell growth and also as biofilms. And independently, they've also assessed the effect of our compounds on biofilm formation. And you can see here that, again, some of our compounds are proving extremely effective at inhibiting biofilm formation by Candida auris. And in the next slide. So what we've done over the last year, even during lockdown, is to confirm that both Candida albicans and auris possess an alternative oxidase, that this recombinant AOX is sensitive to inhibitors that we have developed. And in collaboration, as I say, with our spin-out company, we've developed quite a number of these inhibitors. And the inhibitors themselves have been driven by the crystal structure. And in collaboration with Symcell in Sweden, we've demonstrated that these inhibitors drastically inhibit biofilm formation, in contrast to some of the use of fluconazole. And in my last slide, I just want to show the people that have been involved in this within my laboratory. And in the final slide, then again, welcome you all to Brighton Beach, and you can see here people involved in that once down on the beach, and of course hopefully that will return post COVID-19. And again, apologies for technical problems, but hopefully this has come through. And many thanks to Mark for helping me get through this. Thank you.
Thank you. You showed your professionalism there as a lecturer, I felt. So there'll be any questions, Tony, will be in the chat.
Okay, yep.
And I'll hand back over to Snehal
Yes.
to introduce our last speaker.
Yes, thank you. So our last speaker is Dr. Bharti Prakash, who's from the University College in Mangalore, and she is going to talk to us about her work on denture biofilms. So I'm going to hand it over to you, Dr. Bharti. I think you're on mute. Sorry, we can't hear you. Yes.
Yeah, thank you. Good evening. Thank you for giving this opportunity to share my topic with you. And thanks once again. As the topic has been told, I would like to share the screen, and please inform if you are able to see this. Is it seen?
Yes, yes, we can see the screen.
Can you see this? Yes. Hmm. Denture biofilm. Just now we heard a little bit about the Candida biofilm, and it is a continuation, we can say, but in the different aspect. I teach in the Department of Microbiology at University College Mangalore of Mangalore University. And this work has been done as a part of my PhD work at UNESCO Merson Center at College of Fisheries, Mangalore. As we all know, due to accidents or due to growing age, we may have to replace our natural teeth by the denture set. And it is an artificial teeth set which needs a lot of fabrication and a lot of efforts from the efficient dentist and a prosthodontist rather. And we finally get this. But first time when this has been inserted into the oral cavity, it's very difficult till the person gets accustomed because it is something new, and denture, our oral mucosa takes a lot of time to get accustomed to it. So whatever is this small denture, if I have highlighted one small area, it is a polymethacrylate, but it is of two types: heat cure and self cure. Yeah, but microscopically, if you see, there are many microcapillaries and the channels and the small pores, which later becomes the ideal niche for all the oral microbes to form the colonization and later to the biofilm. And all this biofilm, especially the Candida, it makes a lot of a difference to the oral mucosa. And those who are wearing in the initial stages, they suffer a lot from this erythema kind of pathology. And this is a generally called as Candida-induced denture stomatitis. It's very painful, and for the time they get adjusted, it really needs a lot of fabrication to be done for the denture. By then, maybe within 1 month, this biofilm gets very well set in the denture, and it is a non-shading surface. So definitely lot of not only micro candida, but there are so many other microbes entangled together and forms an ideal biofilm of the oral cavity. And every time it just keeps changing based on the nutrient that we take, based on the hygiene that is followed by the person, and of course the health condition. Now the sample collection was done from the denture wearers who are wearing it for minimum of 1 year, for 1 year period, and we had grown it on the Sabarot agar. It was well isolated. The similar study was also done. Now here for the denture, we had taken then the palatal focal facing surface of the denture was swabbed, and it was grown on the Sabarot's agar. And equally, we took the palatal surface swabs from the non-denture wearers just to have the comparison. So when 100 such denture wearers were observed, we came to know that majority of them were of Candida albicans, and that is almost 60%. And the remaining, like Topicalis, Glabrata, Dubliensis, and Cruzi, they were in the minimal order. So majority here also say we know Candida albicans is one of the oral microflora, normal flora of the oral cavity, but still it is also an opportunistic pathogen, especially for these old people, and if they have such stomatitis condition in them. So comparatively, we have only 2 species that are there in the non-denture wearers, that is normal people with the dental set in them. Now, when I tried this biofilm formation using 96 polystyrene microtiter plate. So all those, when they were tried on using a media called yeast nitrogenous broth. So there are 4 types rather. One was around 40% of them were non-biofilm formers. Some were moderate. Now it is shown over here. These are the strong, and these are medium biofilm formers, and these are the non-biofilm formers. And 40% of them were the non-biofilm formers. The weak is also given, that is intermittent to this one. Now, among these biofilm formation, which was tried for all the 100 organisms, Candida albicans species, it was tried on yeast nitrogenous broth as well as its 1:10 dilution. So with the diluted media also, there was not much difference in their biofilm capacity. So weak biofilm formers and the moderate biofilm formers were more, and the strong biofilm formers were very less, that is 1. But in the diluted one, we got it as a 2%. That means as it dilutes, also the biofilm will set strongly on the surface, and that is also the percentage has doubled even in the diluted condition. Whereas we got in the non-denture wearers, it was only 73%, and in the diluted we have got more. So it is the same case. Non-biofilm formers were more over here, but since they are not in a denture material, this biofilm, that is the reason we have got very no biofilm in this area. All of them, when they were taken, it is a elaborated table which is showing that the 1:10 is also showing us, and this is not only for albicans but the other species also. So their pattern of biofilm formation is seen, but Candida strong biofilm formation has been seen among the Dubliensis and the Cruzi. And when we dilute them, the number is almost the same. So here some of the species have not shown major difference in the biofilm formation. Similar thing we have checked for 1:100, and this was too much diluted compared to 1:10. But here also the pattern of the biofilm formation is more or less. This equally shows that even in the diluted nutrient condition, we get the similar pattern. Even though there is a smaller traces of nutrients, we find the similar biofilm pattern among the same isolates in different nutrient conditions. This in only for tropicalis and albicans, when we compare, almost most of them are, this is for the non-denture wearers, like the normal people without any set. Since there is no such non-shading surface, this has been found quite normal, and only the weak biofilm formation has been seen. But this was tried only with one broth, that is YNB, and its 1:10 dilution. Usually we say that all these biofilms, they are resistant to the these organisms involved in the biofilm formation. They are resistant to most of the antifungal agents and the antibiotics also. And this was also tried using 4 different antifungal agents. So here we see that the resistance was seen at the most to the clotrimazole and very minimal to the amphotericin B and the fluconazole, and least was found for the nystatin. So this it is equally true, but it is not true in case of clotrimazole, and it is not much affected in case of amphotericin B. So any kind of oral topical treatment that has to be given, then we have to see that they are not. It does not involve clotrimazole. And for the non-NDW is non-denture wearers, even they have shown the resistance to some extent, but they are completely sensitive to the amphotericin B, whereas in the test condition we have seen at least 18% of them were resistant, but here we find the complete sensitivity to this amphotericin B. So it has partially shown that biofilm also makes it part resistant to the antifungal agents even in the oral cavity. And these isolate we have completely like separated, and we are testing it outside in vitro. But if it is taken as a whole oral biofilm, that may give a complete resistance, because other than this there are many other microbes entangled in the biofilm, and this film is going to resist the antibiotics which are percolating through the biofilm till the base. And that is the reason, since it is isolated, we are finding slight variation in the antibiogram pattern. Now it was difficult to observe this biofilm in the 96 wells. So what was done, this is another set of experiment where 24 wells microtiter plate was taken, and since the wells are quite broader, we had inserted the glass cover slip which is round, and on this glass the similar experiment was conducted how it was done for the 96 well plate. And there, after this biofilm formation of 1 day is over, we could see and inoculant was taken as a one representative from the strong, one from medium, and one from weak biofilm. So, and the non-biofilm also. And we have seen the one which produces strong biofilm on the glass using calcofluor stain. We have seen it gives a lot of matrix we see around in this diagram. And the one which was producing medium biofilm, the cells are quite loose and separate, and not much of matrix has been synthesized during the biofilm formation. It was for 3 days, and when we take it for this is the same experiment where we had used a
Hi, Dr.
non-biofilm.
Bharti. Sorry,
Yes,
I just wanted to check if you will be able to wrap up in
yes,
a minute
no, it is
because
done.
we're already. Yeah, thank you.
Yes, it is done. Now, these are the 80cc strain we had checked, and that is also giving us this picture. And calcofluor stain and all the biofilm forming stains of Candida albicans, they show the presence of ALS gene in them. And this gene was also targeted, and using PCR and using this ALS gene, we tried to check. It has shown that all the Candida albicans isolates have shown the presence of this gene. That indicates that this gene is present in all the Candida of the oral biofilms. And all the conclusion, finally, I would like to give you that denture biofilms are more, they show the more multi-species of Candida, whereas compared to the non-denture virus. And the biofilm forming ability is found in approximately more than 50% of the Candida isolates, whereas diluted media also shows similar biofilm pattern. So it is not that only the nutrient rich will show and the other vice versa is not true, but even the diluted nutrient media shows similar biofilm pattern. And only the strong and moderate biofilm formers show more resistance to the clotrimazole than the fluconazole and nystatin. Now all this Candida which shows the presence of ALS gene coding for the biofilm formation, but not all of them are giving us the strong or moderate biofilm formation. So the reason for this is so many other factors which are also helping in the natural oral or the denture biofilm, but that cannot happen when we do it in vitro. And this is the reason why the oral biofilm itself is an ideal example for the biofilm to study. So this is just a very small piece of that. Similarly, there are so many organisms, especially anaerobes also, they contribute a lot for the generation or the formation of this biofilm in the denture. So it is a challenge to synthesize a material of the denture which is free from biofilm or which resists the biofilm formation, and that is going to be the solution for those who wear the denture, who can get rid of this denture induced Candida induced denture biofilm and the denture stomatitis also. Thank you for your kind attention. My work has been done in KVG Dental College, and it's a part of the PhD work. I thank NBIC, Mark, and all the participants, India Biofilm organizers, for this webinar and giving the opportunity to present my work before you. If you have any collaborations to further design or fabricate the denture material which is biofilm proof, it would be good. So if you have any suggestions in this way, you can provide. Thank you for your patient hearing. Thank you so much.
Thank you, Dr. Bharti.
Thank you, Dr. Bharti, for your wonderful presentation. So thank you all very much.
Thank you.
Joining us today. We're very grateful for the audience putting aside their Saturdays to hear our wonderful set of presenters. I'd like to thank all of them for their concise and clear presentations and for putting the time and effort into preparing them. And on behalf of Will and myself, I'd like to say thank you from the National Biofilms Innovation Centre. Just to remind you that we will be sending the recordings, a link out to the recordings to all presenters and attendees within the next week. I'm going to hand over to Karishma to close the meeting and to thank her and Snehal for involving us. Thank you.
Thank you, Mark. And well, thanks to you and the bigger team at NBIC for always being so responsive to our suggestions and ideas. We look forward to taking this further. I know we've applied for a few things, and let's see if they work out. It'll be wonderful if they do. Moving back to India, I think all roads have led to UK for some reason. After 12 years in the United States, I knew nothing and no one in biofilm research in UK, and now suddenly I only seem to be heading in that direction. After our meeting at Glasgow in 2019 with Snehal. So I have to say a special thanks to Snehal for taking this forward from India Biofilms. Now she's actually bridging India and UK biofilms, if you want to call that. Our loss is definitely your gain, right? So Snehal starting her PhD in the UK, and she just left my group in India. All right, so I often talk of India's brain drain. I haven't been able to stem it myself, but hopefully we will entice her to return someday. And thanks to everyone. Thank you to the panelists and the attendees, and we look forward to taking this further. Thank you.